Oxford Biomedica is a quality and innovation-led viral vector CDMO with a mission to enable its clients to deliver life-changing therapies to patients around the world. One of the original pioneers in cell and gene therapy, Oxford Biomedica has more than 25 years of experience in viral vectors, the driving force behind the majority of gene therapies. Oxford Biomedica collaborates with some of the world's most innovative pharmaceutical and biotechnology companies, providing viral vector development and manufacturing. Oxford Biomedica's world-class capabilities span from early-stage development to commercialization. These capabilities are supported by robust quality assurance systems, analytical methods, and depth of regulatory expertise. Oxford Biomedica is headquartered in Oxford, UK. It has locations across Oxfordshire, UK, and near Boston, Massachusetts, United States of America. Now, it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Nick Clarkson. He is the Vice President, Head of Platform Research at Oxford Biomedica, which drives innovation of Oxford Biomedica's core Lentiviral platform through vector design and improvement, generation of packaging and producer cell lines, small-scale process development, computer-aided biology, and development of core analytics. The group's mission is to keep the Lentiviral platform at the cutting edge of gene therapy and to ensure its applicability to the needs of the wider cell and gene therapy community. Nick obtained his PhD in molecular immunology from the University of Oxford. And next, it's my pleasure to introduce you to Dan Farley. He is the Senior Director of the Vector Engineering Group. Oh, there's Nick. Hi, Nick.
Hello, everyone.
Hi. Now it's my pleasure to introduce you to Dan, and there's Dan. Dan Farley is the Senior Director of the Vector Engineering Group at Oxford Biomedica. He joined the company in 2004, having completed a PhD at Warwick University working on adenoviral vectors and cell line development. The Vector Engineering Group was founded in 2013 to improve OXB's lentiviral vector platform and technology for cell line development, which later expanded to vector-agnostic technologies encompassing AAVs and adenoviral vectors. Now, it's my pleasure to pass the mic and the controls over to our first speaker, and that is Nick. So Nick, when you're ready, you may begin.
Thank you, Sonia. It's a pleasure to be here, and thank you all for joining us for this XTalks webinar, where we're going to talk a little bit about TetraVecta and lentiviral vectors in general. What we really hope that you're going to learn today is some of the advantages of using lentiviral vectors in cell and gene therapy, how you can improve titers, and we're going to show you some of our novel technologies that we've developed. We're also going to talk a little bit about some of the important aspects that can derail or delay product development, and this can be anywhere from particle quality, interactions of your gene of interest that may affect processing and yields. Also, some of the big questions like transgene cargo capacity, because it's always an issue in cell and gene therapy, and also talk a bit about the potency, and this is potency of your gene of interest expression in your host cell and how this interacts with the host. And finally, we'll hopefully learn some of the novel tools, including our 4th generation lentiviral vectors that are actually being designed to address some of these needs and issues that are encountered. So this is the agenda today. I'm going to give a brief overview of Oxford Biomedica, a little introduction to lentiviral vectors, and I'll hand over to Dan, who's going to take you through the 3rd generation and 4th generation LVs, and we're going to finish with a summary and a question and answer session. So I'm just going to give a brief introduction to Oxford Biomedica. Sonia had said some very nice words about us, but we've been around for over 25 years, and we have a broad range of experience in viral vectors. Everywhere from lentiviral vector, which we initially started with, to adeno-associated viruses, and also adeno manufacturing. And to date, we've made over 340 successful GMP batches in the last 10 years. This is mainly due to our client base. We are international. We have 24 global clients, 41 partner programs, and our lentiviral vector platform is approved with products in over 40 countries. What really makes us successful in this, it's our end-to-end services for all the vector types, everywhere from construct and plasmid design, analytical development, all the way through process development, manufacturing, QA release, and stability studies. What's really underpinning this is our proprietary platforms that we can offer in AAV, lentiviral vector, and adeno. This is really backed up with our quality systems, expertise, and our state-of-the-art facilities. And in addition to our proprietary platforms, we also have the ability to tech transfer in other platforms from outside of the organization. So really, our success is based on our innovation, and we have what we call our innovation engine, and this has delivered multiple breakthroughs, particularly in the LV technologies and now in the AAV field. We spend a lot of time looking at cell and vector engineering. This has really gave birth to our next generation lentiviral vectors that you're going to hear about shortly, the 4th gen vectors, but also producer cell lines, which is something that we've really excelled in the recent years. We innovate a lot in the large-scale process production, and this has led to new platforms using perfusion for a lentiviral vector. And this is perfusion with certain enhancers that we're going to talk about. This boosts your titer and quality. But we're also very keen on the analytics and data. So we maximize our use of data and integrate it. And this leads to us using a lot of design of experiments to optimize plasmid ratios and transduction and other parameters. And we use things like proteomics and transcriptomics to characterize our viral vectors, them being very complex biological entities. You need some quite advanced techniques to understand them and characterize them. And then moving forward, we actually have automated a lot of our assays going into QC release, which gives you the precision and consistency that's required for vector production for human use. So I'm going to give you a brief introduction to lentiviral vectors. Now, lentiviral vectors, they're well known in the industry, and they're particularly good at delivering large payloads, and they have an excellent safety profile and long-term gene expression. Now, the most commonly used lentiviral vectors for gene therapy is typically based on an HIV-1. This is what we most commonly use in our platform, and it has the ability to stably integrate into the host genome. It's particularly good because it's non-pathogenic, which Dan will talk about in a minute, and there is a lack of pre-existing immunity in the general population. It can deliver genes to both dividing and non-dividing cells. It actually does this in a very efficient way, which it enters the cells, it protects the cargo, so it protects the RNA, delivers it to the nucleus. You only need one integration event that would happen, and it's stably integrated into the host genome, and you get the effects of the gene expression. And the fact that it does this really means that it avoids a lot of the innate responses, it avoids a lot of the nucleases and base modifications that can happen as part of host natural defense against viral vectors. So it makes them really useful for permanent modification of cells. Has the other advantage of delivering up to 10 kb of transgene capacity, and it's now actually being considered for in vivo high dose applications, and this is being made possible through innovation in envelope retargeting. Traditionally, it was antiviral vectors have been used ex vivo. So as I was saying, they've been widely adopted in the cell and gene therapy, and there's 9 commercial products to date, and they actually make up about 43% of ongoing clinical trials in cell and gene therapy. And the biggest market really is in oncology. Everyone is probably aware of the CAR T and the amazing results that have been achieved by using modified T cells in oncology, and the majority of products now moving forward in the CAR T are based on lentiviral vector gene delivery. So I'm now going to hand over to Dan, and Dan's going to take you through some of the aspects of the 3rd generation lentiviral vectors, and then talk to you about some of the improvements made and the 4th generation vectors. So over to you, Dan.
Okay, thanks, Nick. So as Nick said, I'm going to just give you a little bit of introduction in terms of state of the art, and then we'll move on to TetraVecta and what that means. Okay, so this slide just gives you an overview of third generation lentiviral vector components. So it's a 4 plasmid system, and I'm highlighting here the kind of main takeaways from this, right? So focusing on the genome plasmid, third generation vectors are driven by a powerful promoter that drives the vector genomic RNA that gets packaged. And this is important from previous generations because that happens in a way that's independent of an auxiliary protein called TAT, which was seen as an important safety feature. I'm just highlighting a couple of key cis elements in here. So sequences that are retained in parts of the sequence that are kind of legacy sequences from HIV, from which the vector was derived. And that includes the packaging region, which contains this major splice donor site, and the reasons why we're highlighting that will become obvious later. The Rev response element, so this element is necessary to be acted upon by Rev, which allows you to generate packageable RNA. And third generation vectors also employ what are called self-inactivating LTRs, and this removed promoter activity and was also seen as an important safety step. The actual transgene cassette relies on the polyadenylation sequences that terminate transcription of the gene of interest mRNA in the target cells. And as we'll go on to later on, we'll talk about the relative strengths of these sequences. And in production, we typically include a backup poly-A in there, and reasons for that will become obvious later. The WPRE, which is an element typically included to increase expression in target cells, is also one of the main features of third gens, and we employ a mutated version of that. Okay, so and then moving on to the packaging plasmids, we have 3 of those components. So that includes the GAG-POL cassette, which we code and optimized, and that has the effect of both minimizing homology between some of these components to greatly reduce the ability of these components to recombine and potentially generate something that's infectious. The other consequence of that is that the GAG-POL plasmid is no longer Rev dependent. So typically historically most GAG-POL plasmids also have an RE element, as they do in the genome, and that's dependent on Rev. So obviously that doesn't happen in our system. And again, that will be the reasons for highlighting that will become obvious later. And then we're obviously co-expressing the Rev cassette, and then you have the ability to pseudotype lentiviral vector particles by supplying any kind of viral spike protein, but typically the field has used VSVG. Okay, so we wanted to just pause briefly before we moved on to TetraVecta to talk about some of the technology that we have developed to just increase the output of lentiviral vectors in general. And one of those is called U1 technology, and this is essentially a U1 snRNA molecule, which is a non-coding RNA which is normally involved in splicing in all of our cells. And what we've done here is we've taken part of the sequence here that is on that U1 snRNA, and instead of being able to involved in splicing, it can now bind to the vector genomic RNA. And in doing that, in co-production with all the other components we've just described, we see a kind of a dose response. So, you know, the more of that plasmid we put in, the higher the titers we get. And we see anywhere between a sort of a 2 and sometimes we see up to 10-fold increase in titer. And the main reason for that is because we are stabilizing the pool of packageable RNA in the cells, and therefore you're actually able to fill more of the particles that you're generating, and that has the effect of improving P to I ratio. So here, the lower the number of particles means that more of those particles are active. And so we can talk about the fact that, you know, this technology is being used by some of our clients in GMP to great benefit as of today. The second technology we want to talk about is our U2 technology, which is a class of protein kinase C agonists, and we've essentially found that these are able to give the cells a bit of a kick during production, and it increases expression from many of the promoters that we're employing to drive expression of a lot of those components. And this particular version, which is called Ingenol 3 alginate, we can see that we're able to see some quite significant increases in titer, and again also an improvement in this P to Y ratio, so that the specific activity of the particles we're generating is improving. And we have used these two technologies together to get combined effects, and we've done quite a lot to show that that these inducers are cleared, and in the case of U2 down to undetectable levels. And so these two technologies we recommend also combining with our TetraVecta system as well. Really, we wanted to kind of highlight some of the experiences we've had in developing lentiviral vector manufacturing processes for our clients and obviously for ourselves. And sometimes this can seem a bit like a black box. So, you know, we have our gene of interest, we select maybe some variants and do some optimizations, and then we may instantly be thinking, great, you know, let's just get some Lentiviral made, it's all going to work out fine, and we'll be off and away in a clinical trial. And really we want to highlight that, and obviously an aspect of that is as long as we get the right yields, everything's fine. And it's been our experience that the transgene and Lentiviral design can have a significant impact on the success steps through this pathway to actually realizing you know, a batch that you're happy with, and going into a whatever kind of trial you'd want to perform. And so, you know, taking some of these one by one, you know, right from the word go, I guess one of the questions we'd be asking people is, you know, have you already been able to onboard your favorite candidate gene of interest cassette, or have you already had to make a compromise because you have had an issue the moment you onboarded onto the genome, you didn't make enough titer? One general thing we would ask people is, you know, do you know the source of your Lentiviral cassette? You know, we have some experience with some very old legacy genomes with clients coming in and not quite understanding, you know, what's happening to their cassette. We're also aware of widely available lenti genome plasmids that have significant open reading frames from HIV left in some of these backbone sequences. So, you know, that's a good question to ask yourself. Do you understand the vector you have in your hands? Then there are other questions, you know, sequence optimization, do we code and optimize? What about splice sites, repetitive sequences? You know, maybe the size of payload, are we having to compromise on what we onboard because, you know, the titers tend to go down the bigger the payload? And then also probably one of the most significant aspects is your gene of interest expressed during production? And, you know, is that impacting on your upstream titers? Is that gene of interest secreted or a membrane protein? So is it ending up in the feed that's going into your downstream process? And then there are, you know, potentially knock-on effects. You know, what are some of these consequences with, you know, quality of the final product? Have you actually got protein in your final product? And we'll come back and touch on that. And obviously, there's all of this above can potentially impact on the success of this pathway. And so we're arguing for a kind of a LV design centric view, spending a bit of time here, getting things right before you move through this pathway. So now we're going to go on and talk about how we think the TetraVecta simplifies what potentially is, you know, a complicated process. Okay, so what we've really done with TetraVecta, sort of playing on the whole idea of it being a 4th gen, and 4 aspects. So most people agree, it sounds like from that poll, that obviously people believe that all these aspects are important. And so we're going to take you through these 4 technologies that will address one or all of these. So firstly, that's the 2KO genome, which forms really part of the core of what TetraVecta is. We're going to talk about the TRIP system and how that works better in our TetraVecta system. We're going to talk about max packs, which is more kind of tip of hat to the capacity aspect. And then we're going to finally talk about our super LTRs, which is really a nod to kind of safety and potency expression in the target cell. Okay, so the first really technical slide on 2KO, as I said, this is really the core of TetraVecta. And so taking you through here, we've got a schematic here showing of what the kind of RNA is actually generated from a third generation expression cassette during production. And so obviously, ideally, there's the full length RNA that which you want to package. And usually, if your internal promoter is active, you will also get some expression and translation of your gene of interest. But what we, and also you can find in the literature, have discovered that there's a lot of aberrant splicing happening, and that is happening from this splice donor that's embedded in the packaging signal of that's derived from HIV. And so what happens is you get bypassing of this RE element even in the presence of Rev. So the dogma is that Rev will bind to the element here and stop this from happening. And so the consequences are that you make this truncated RNA, and you will get some gene expression from here. And so this also goes some way to explain why perhaps some people might have a tissue-specific promoter that they don't think is active in these sort of 293 base cell lines. Say, they might take that cassette from an adeno or an AAV cassette and they put in a lenti, and suddenly their tissue-specific promoter lights up the cell. And really the reason for that is this splicing activity. And so we can see that in this RT-PCR, we can see that we have total RNA here in the cell, and what you can actually also detect in the virions. So we can actually detect these spliced products in virions. Okay, and so what we've essentially done is we've been able to mutate and to ablate all this splicing activity from our 2KO genomes, and that leads to very clean production of full-length packageable RNA in your production cells, and that obviously translates to a single species being present within the vector particles. And so during the course of our studies, we found in doing this, you actually reduce the titers. And so we're able to rescue those. The solution to that is by rescuing the titers with our Rev-dependent version of our 2K genomes with our U1 technology that I've described previously. Okay. So I'm going to move on to talking about TRIP system. So our TRIP system was actually developed a number of years ago now, and it's a translational repression system that stops your expression of your transgene only in production, and that happens by a co-expression of a protein called TRAP, and that binds to its target sequence in the RNA that's placed immediately upstream of the primary start codon of that protein, and so you stop the ability of that mRNA to be translated. And so what we're able to do here is to stop the side effects of your gene of interest, and that might be some obvious side effects like cell viability, or perhaps some less predicted things like on assembly, which might impact on, say, your upstream, your output, the amount of vectors actually coming out of your cells. Even if that doesn't impact on titers, as I said before, you can have issues with your gene of interest being shed into the feed or even actually embedded within your vector particle, and that might have some consequences downstream in downstream purification. Okay, so we've generated some sequences now that can kind of plug and play into lots of different settings, and we've demonstrated that for both Lenti and across some other viral vectors as well. Okay, so the TRIP system was developed really to kind of park a lot of these issues with sort of specific to the gene of interest you're employing. Okay, and then so what the issue is with the third gen vectors is that some of those spliced vector genomic RNAs that can encode transgene are poorly repressed in the TRIP system. So what we're able to do is partner both the TRIP system into our 2KO genomes, and we're able to get a lot better levels of repression. And that's leading to significant increases in titer where we know that the transgene is having a problem. So in this case, there's a chimeric antigen receptor gene of interest for CAR-T where we know the titers were very low, and then by employing TRIP on a 2KO genome, we're able to see both obviously the expression of the actual CAR, and which is demonstrates that TRIP doesn't have an impact on your target cell because obviously TRAP's not there. And integration is another reporter on the amount of vector we're producing here. Just one little comment here is to say titers are a relative term, and maybe we can touch on this in the Q&A. So because people use different assays to determine their titers, it's not very helpful to try and compare between different platforms. So we probably should pick that up in Q&A. And then, so this is what's called a Western blot. This is showing the level of protein both in the production cells above here. So in this case, when we're applying the EF1 alpha promoter, this is poorly repressed. And in our 2KO genome, we're able to see really nice levels of repression relative to the third gen. And we're also able to demonstrate that this stops CAR from entering into your particles. And so perhaps for something like CAR, this could be really important. So for example, if your CAR perhaps might target your lentiviral vector to a B cell, you might not want that to happen because overexpression of your CAR in a B cell, i.e. your target cell for CAR-T therapy, might downregulate the antigen. And so you might get some kind of pseudo-negative relapse in that indication. So TRIP is a really nice way of really kind of downregulating gene of interest protein within your press. Okay, so I'm going to switch tack again and talk about MaxPaks. So I realise this is a little bit complicated, this slide, but what we really wanted to do here is to show you the kind of two flavours of genomes that we are offering under the TetraVecta platform banner. So I previously talked about what we're calling 2KO, it's super 2KO. So because of this mutation where we require the U1 enhancer during production to give us maximal titers. And during the course of our studies, we were looking ways of being able to make 2KO genomes without U1. And essentially, the way we've done that is to replace the RE, which is normally acted on by Rev, with a synthetic vector intron, what we call a vector intron. And so what's happening here is that we're just splicing out a little synthetic intron, and that's leading to stabilization of the vector genomic RNA. Okay, so there are two subtle ways in which we're applying these. So the caveat with MaxPaks genomes is because we're stimulating splicing, it means that your gene of interest or your cassette that you're trying to deliver to patients can't have an intron because those will be lost. And so the reason we're offering these two options is if you really want an intron in your cassette, you would use this 2KO genome, and if you're not bothered, you would probably go for a MaxPaks. And to be honest, you know, for indications where you're trying to cram as much possible into your cassette, you're probably already thinking about minimizing some of these cis-acting features like introns to try and squeeze in as much into your payload. Okay, so the other thing I should say is because we're using, going back to what we were saying earlier, we use a codon optimized GAG pol that's Rev independent. So that means we don't actually need Rev in our system. And so for MaxPaks, we're able to minimize down to 3 plasmids, and we think that you know, that will significantly help you know, the optimization around production of MaxPaks vectors. So here's a little bit of initial data which, you know, is interesting, showing that, you know, when we can generate a bit more space in the backbone, we're able to see some stimulation in titers. And so, because obviously the intron itself doesn't contribute to splicing, okay, so we've been able to remove the RE. We've also been able to remove some other aspects of the patching signal to give us about a kb's worth of extra space here. Okay. Okay, and then I'm going to talk, to take you through a couple of slides on our super LTR. So super LTRs stand for sequence upgraded poly A LTRs. Okay, so going back to our sort of comparison to third gens, so this self-inactivating feature that was put into third gens, it resulted in deleting these promoter activity sequences that we don't want, but it also had the effect of deleting poly A sequences, enhancers that we do want. And so the consequences of that is that the poly A's that are used by the transgene cassette are weak. And so there is reading out, there's transcriptional reading out of an integrated cassette, and there's also potentially transcriptional read into the cassette. Okay, so there may be some position effects that happen on your internal cassette you might not want. Okay, and obviously if your third gen has a major splice donor, then it might partake of some kind of splicing out, either into the cassette or outside. And there's been some publications on this happening in the literature. Okay, for the super LTRs, we've essentially put back in enhancer elements in a way in which we've not obviously put back in the promoter activity, so it's still a SYN in the traditional sense, but it harbors these much improved polyadenylation sequences. And we've also been able to provide transcription insulation on the bottom strand, so we're able to stop any transcription coming through on the bottom strand as well. We've seen a stimulation in the level of gene expression as well, which I'll go on to show you now. Okay, so I'm going to take you from left to right. So here's just a compact bit of data showing a comparison where we've just got a luciferase reporter here in our production cells, showing what happens if you just don't have any termination at all, which is that kind of a control. That's compared to a SYN LTR, and then that's now comparing to our optimized. So we're seeing a pretty significant increase in poly A activity in these poly A's in these super LTRs. And then when we look at an integrated Lenti, comparing both third and 2Ka super LTR combo, we're again seeing something like a 50-fold reduction in transcriptional read-in from cellular promoters into our Lenti cassette. Okay, and then on the third panel here, we're showing some data across different cell lines using different promoters, showing that we're starting to see a consistent, albeit modest, but probably helpful improvement in transgene expression, somewhere in the region of 2 to 3-fold on average. Okay. And then this is kind of putting it all together, just our summary slide, comparing, putting all these things together. As I said, these are kind of these are 4 aspects that we can kind of potentially put in modular genomes, so you can pick and choose what you want depending on maybe whether your gene of interest is out giving you an issue or not. But it really just kind of gives a summary of what I've just been talking about. So we ablate this aberrant splicing. We no longer see these splice products in particles, and we have the ability, if we employ the TRIP system, to also stop our transgene of interest being embedded within the feed. So we think that this would potentially translate to cost savings through downstream processing, and potentially shortening that development time as well. And as I said, I'll just give you an idea of what the super LTRs can do in terms of transcriptional insulation. Okay, and then coming on to really what my last technical slide. So particularly for chimeric antigen receptor produced cell lines, it's been found that really in transient, at least, it's typical for those CARs not to show any kind of acute toxicity problems. You're only manufacturing over a number of days. That's not to say that we haven't found particular CARs where the TRIP system benefits, but I would say generally, it's not impossible to make CARs in transient. But when you move to a stable cell line setting where your components are integrated into your producer cell line, you're then talking about having to keep those cells going for a long time through the banking process, and ultimately when you're actually building up a seed train to into your GMP manufacturing, what we really find is that these CARs are actually quite chronically toxic, and that's had real implications on the ability to generate producer cell lines for CARs. And so we've been able to combine various aspects, the main 2 being the 2KO and the TRIP system, to really shut down or minimize expression of CAR. And that means really you get a bigger pool of cells coming through to screening, and you're much more likely to pull out clones that are going to be generating high titer vector. So this has really been kind of a breakthrough moment for producer cell lines. So yeah, and obviously it's not specific to cars. This is potentially specific across the board. So yeah, with that, I shall hand back over to Nick.
Great, thanks, Dan. Control right. Okay, perfect. Yeah, so thanks, Dan. That's quite an in-depth summary of our TetraVectors, but I really hope everyone took the key take-home messages from this. I mean, the first being we can produce higher quality, higher titer vector, with the added benefit of more potent lentiviral vector particles using the TetraVecta. And this really leads into, it allows for the successful manufacture of previously non-viable LV-based products. And we mean this by, when you look at the commercial aspects of producing a viral vector for a therapy, if your titers are very low, it may not actually be a viable product in the long run. But some of these technologies, some of the enhancers, the reduction of the splicing, maybe the trip system, you can actually significantly increase your titers and make it a much more attractive proposition to produce this vector and use it to treat patients. The added ability of onboarding large or multiple transgenes is an added benefit. There is a trend in the field now, instead of just sort of monocars, we're looking at dual cars or multi-cistronic cassettes in the lentiviral vectors, and this is just simply because science is moving on and there is more need for sort of checkpoint inhibitors, multiple targets that people are looking to address in oncology. And again, the development of high titer stable producer cell lines, this has been a real issue in the field. It's successful for some products, particularly with lentiviral vectors, but there's always been a problem with the chimeric antigen receptors, be it due to toxicity or other splicing issues. But using the TetraVecta with the trip system, we're now able to develop these and match or surpass titers for transient systems. And this really paves the way for really big indications where you're going to need a lot of vector and very consistent in its production as well. And this consistency really leads in as well to in vivo applications. Now there is a movement in the field to start exploring the possibilities of, say, in vivo CAR therapy, where the vector's directly injected into the patient. The adoption of these TetraVectors, like Dan had highlighted, the improved safety with the super LTRs, the use of the trip system to stop any transgene being expressed and incorporated into the vector, which may result in off-target, and also the producer cell lines. And this should greatly improve the quality of the vector, the consistency, reduce residual DNA. So I hope you really enjoyed the vector, and I hope you've learned a lot from it. There was a lot of molecular biology, but this is recorded, so you can always go back and re-watch it, or alternatively ask us questions in the Q&A. We're also happy to talk to anyone from research or industry that are looking to evaluate the TetraVecta of any of these components, and we can be reached at the partnering@oxb.com email address. So thank you all for listening, and we're happy to take questions.